Welcome to this module on membrane dynamics and protein-mediated transport, where we'll explore how cell membranes move, adapt, and control the movement of molecules. Over the next slides, we'll break down membrane structure, the proteins that mediate transport, mechanisms like endo and exocytosis, experimental tools, and the biomedical implications of these processes. This slide outlines our roadmap. We'll start with membrane structure and dynamics, then examine protein transport mechanisms, vesicle trafficking, and regulation by signaling pathways. After that, we cover experimental and computational methods, disease links, therapeutic strategies, and emerging technologies that push this field forward. In this overview, we define biological membranes as thin, flexible barriers built from a lipid bilayer and embedded proteins, and highlight why fluidity matters. The images show typical membrane organization and the concept of a fluid mosaic, helping us appreciate how structure supports selective permeability and protein activity essential for cell life. Here we summarize key membrane components and their roles: phospholipids and cholesterol shape, bilayer properties and fluidity. Integral and peripheral proteins provide transport and signaling functions, and carbohydrate chains contribute to cell recognition and protection. Together, these elements create a balanced surface that supports complex cellular behaviors. We'll review membrane models that explain how membranes behave, from historical sandwich and lipid protein ideas to the modern fluid mosaic model. These frameworks help us understand how proteins and lipids move within the bilayer and why those dynamics matter for transport, signaling, and membrane remodeling in living cells. Membrane dynamics have clear functional consequences. They influence signaling, transport efficiency, and overall cell function. Fluidity depends on hierarchical factors from lipid chemistry to temperature and protein interactions, and small changes in composition or temperature can dramatically alter membrane viscosity and the behavior of embedded proteins. Lipid mobility is central to membrane behavior and occurs through lateral diffusion within the plane, rare flip-flop between leaflets, and rotational movements. These motions are regulated by temperature, lipid composition, and interacting proteins, and together they allow membranes to reorganize quickly in response to cellular needs. Membranes can undergo phase transitions, shifting from a more ordered gel state to a fluid-liquid crystalline phase when heated or when lipid saturation changes. These transitions directly affect permeability and protein mobility, and cells exploit phase behavior to tune membrane function under different physiological conditions. Membrane asymmetry arises because lipids are distributed unevenly between inner and outer leaflets, and this imbalance is actively maintained by flipases and scramblases. Proteins that regulate asymmetry influence signaling and stability, and asymmetry changes can serve as functional signals during processes like apoptosis and coagulation. Membrane curvature and remodeling are driven by lipid composition, protein scaffolds, and mechanical forces. Curvature is essential for vesicle budding and fusion during transport, and proteins like bar domain factors help generate and stabilize curved membranes to enable efficient cargo trafficking and membrane reshaping. Transport proteins come in distinct classes with different mechanisms. Channel proteins form pores for rapid selective diffusion. Carrier proteins bind substrates and change shape to move them. And ATP-powered pumps use energy to move molecules against gradients, all working together to control intracellular environments. Facilitated diffusion relies on proteins to move substances down their gradients with specificity. Carrier proteins bind and undergo conformational cycles, while ion channels open and close in response to stimuli to allow selective ion flow. Unlike simple diffusion, these processes provide controlled passage and rapid response to cellular signals. Active transport uses energy to move molecules against concentration or electrochemical gradients. Primary active transport directly uses ATP to power movement. Secondary transport taps gradients set up by primary pumps, and electrochemical gradients are central for driving nutrient uptake and maintaining membrane potential for many cellular functions. Protein structure underpins transport function. Pore size and binding site geometry determine selectivity. Conformational changes drive transport cycles, and structural studies reveal how ion channels and carriers are built to move specific molecules. Understanding these shapes helps explain both normal activity and how dysfunction arises. Endocytosis begins with ligand recognition and receptor clustering, followed by membrane invagination and vesicle internalization. It includes pinocytosis, phagocytosis, and clathrin-dependent pathways, and plays crucial roles in nutrient uptake, receptor regulation, and immune defense by selectively bringing external cargo into the cell. Exocytosis coordinates vesicle trafficking, docking, and membrane fusion to release cargo outside the cell. Snare proteins mediate the final membrane fusion step, ensuring efficient content release, and this process is especially important in neurotransmitter secretion and hormone release, where timing and precision are essential. Membrane recycling restores surface area after fusion events and maintains composition through retrieval pathways. A dynamic balance between endocytosis and exocytosis controls membrane turnover, while recycling routes and sorting decisions determine how transporters and receptors are redeployed or sent for degradation. Vesicle formation depends on specialized proteins. Coat complexes like clathrin, CoP, and CoPI shape budding membranes. Adapter proteins link cargo to coats, and coordinated assembly ensures cargo selection and successful vesicle scission. These coordinated steps are vital for targeted delivery and maintaining internal organization. Transport proteins are regulated rapidly and precisely through allosteric changes and phosphorylation, while shifts in membrane potential and pH alter activity and driving forces. Feedback loops then adjust transport rates to stabilize cellular conditions, creating dynamic control over nutrient fluxes and ion balances. Signaling pathways profoundly affect transport by triggering receptor cascades that recruit messengers like CAMP and calcium. These second messengers amplify signals and direct where transporters are localized, helping cells move proteins to the membrane or adjust activity in response to environmental cues. Cells respond to transport changes by adapting uptake rates to nutrient availability, modulating transporter activity under stress, and coordinating transport with metabolic pathways. These adjustments preserve homeostasis, protect against damage, and optimize resource use when conditions fluctuate. Transport proteins are constantly turned over to maintain function. Ubiquitination tags proteins for lysosomal degradation, while endosomal recycling restores others to the surface. This cycle of degradation and recycling ensures balanced transporter levels and supports cellular homeostasis under changing demands. Experimental tools let us measure membrane dynamics directly. FRAP tracks fluorescence recovery to quantify mobility. Electron microscopy provides high-resolution structural context. And single particle tracking follows individual molecules to reveal heterogeneous behaviors, together offering complementary insights into membrane movement. Studying transport proteins uses methods tailored to function. Patch clamp captures ion channel currents in real time. Radio-labeled uptake assays quantify substrate transport, and inhibitors or mutagenesis help identify mechanisms by perturbing protein activity and mapping critical residues and domains. Imaging and visualization tools enhance dynamic and structural studies. Advanced light microscopy enables live cell tracking of proteins. Fluorescent tagging allows real-time localization, and cryo-electron microscopy reveals high-resolution structures of protein complexes, all combining to link motion with molecular architecture. Computational approaches extend experiments by simulating membrane behavior at multiple scales. Molecular dynamics reveal atomic-level interactions. Data integration techniques combine simulations with experiments for deeper insight, and docking or pathway predictions help forecast protein binding and transport routes. Membrane transport underlies cellular homeostasis by maintaining ion gradients and pH, facilitating nutrient uptake and waste removal, and regulating cell volume and shape. These transport functions are essential for metabolism, signaling, and survival, and disruptions can quickly impair cell physiology. Transport dysfunction links directly to disease. Channelopathies arise from faulty ion channels. Transporter deficits impair molecule movement, Altered membrane properties contribute to cancer progression, and failures in neuronal transport are implicated in neurodegenerative disorders like Alzheimer's and Parkinson's. Recent advances in membrane biophysics include nanoscale mapping of membrane domains, engineered synthetic membranes that mimic biological function, and integrating systems biology to understand complex cellular behavior. These innovations expand our ability to study and re-engineer membranes for research and application. Finally, the field faces challenges and opportunities. Membrane environments are highly complex. Translating basic research to clinical therapies remains difficult, and interdisciplinary collaboration is essential. By combining techniques and perspectives, we can overcome obstacles and unlock new innovations in membrane transport science.